thanks everyone. Thanks for the nice introduction. I also realized I have to update my bio. That was an older version that I clearly submitted here. Thanks for the organization, for the nice event. Thanks too for the invitation. So where are my buttons? Where do I click?
Keyboard just on it.
Okay. Thank you. So for today's talk, I'm going to give a bit of an overview of the things you can do with metabolomics and multi-omics data. And the reason why I have this Pan-Cohort title in here is because one aspect that I'm trying to highlight across all of these different topics is what you can do across different cohorts when you look at reproducibility, replicability, and later on maybe even different species that we compare. So there will always be an aspect in all of these things that I'm going to show today of how to analyze this in different data sets. I'm going to start with something, see the mouse, right? Yeah. I'm going to start with something a little more technical, sort of more the computational work. That's the part where I actually come from. And then I'm going to go through, and as these are very different topics, I have to stay a little high level, of course, given the time, different studies of Alzheimer's disease, and cancer. That's what I actually work on. COVID is what we kind of had to work on. And then a, no pun intended, pet project in the end, where we look at dog metabolomics. So the four bottom ones will be a little more clinical. The first one is a little more technical, but bear with me because there's interesting information in it, I believe. So I'm going to start with that part, just to warm up, I guess. So one thing that was still part of my PhD work, and I'm also showing this sort of from my biocrates angle here. This was actually P150 data from the Cora cohort back then, one of the biggest studies. So we measured in a couple thousand samples, which in the late 20s, so the 2008, 2009, was a big deal, a couple of thousand samples. And what a lot of my research was about are these networks. So what this shows, and I'm kind of just keep it simple, not too technical, is each one of those dots is a metabolite. And when they're connected, that is means they are correlated across samples. So if one is up, then the other one tends to be up as well. It's a very special type of correlation method. I'm not going to go into the detail here. But what the main part of my research was back then was to prove that these things are not just pretty to look at, because they're always pretty to look at, which is a fallacy, actually. I can also generate this from random data. It will still look great. But to prove that if you statistically infer links between metabolites from blood metabolomics data, that these individual links that you see here, all these connections, actually do correspond to real biochemical pathway reactions. So even though the statistical method knows nothing about biochemistry, pathways, or anything, if you do this, what comes out are real pathways. Back then, this was how I had to learn about phospholipids, because I didn't know much about it, but that was what the platform covered. And then over the years, we repeated it on different platforms, integrated with genetics data. But it was just this fascinating finding, because again, this is blood data, right? So you take a blood specimen, and it turns out that the wiring of the metabolites inside of this blood sample is still has so much of the pathways that originally worked on them in the cells, of course, not in the blood, that you can reconstruct the wiring of the network. And for me, this is always a fascinating find, because that's pretty good news potentially for blood-based biomarkers, blood-based interpretations, because the stuff that happens in the organs and the cells still makes its way into the blood, and we can see it to the point that we can accurately reconstruct these pathway cascades. So they're not just pretty to look at, they actually have a lot of information in it. So this was the sort of back in the days. And then sort of one thing we worked on now, this a little more recently, is, so it's a deep learning method, which is called variational autoencoders, which is from the AI field. There's a lot of fancy words, but basically what it means is it's asking, if you're measuring a couple of hundreds or maybe thousands of markers in a data set, do I actually need all of those markers? Are there really 1,000 different degrees of freedom in my data, or can it be condensed? Very similar to file compression on a computer. When you compress a file down, it removes everything that's not needed and sort of gets it down to its core. What it does in principle is it takes the data and presses it through a bottleneck. It has to go down in information and then sees if it can still reconstruct it. And it turns out if you do that, you really reduce, in this case, metabolomics data to what's actually important. Very similar to principal component analysis, PCA, just it has a bunch of advantages of nonlinearities. So what we did is we took a data set that then at that time when we started this project, a couple of years ago, was big. Several thousand blood samples measured with mass spectrometry from the Twins UK cohort, predominantly female, as you can see. It's just a quality of this cohort. Pressed it through this bottleneck, and then applied what we found, these signatures, so the different pathways that you now see in this reduced version of the data, not all of the samples anymore, but the reduced version, to completely different data sets. So we had a Qatar-based metabolomics data set of partially untreated type 2 diabetics that were recruited actually at a dermatology department waiting room. Of course, from a completely different ethnicity, we have acute myeloid leukemia cohort from America and a schizophrenia study also from Qatar. So very, very different studies. But what happens is if you now take these new compressed version of the metabolomics data set, which corresponds to bigger groups, like only the amino acids, only the energy metabolites, only the lipids, and you associate it with the disease phenotypes in these cohorts, they actually show stronger correlations than anything else. So what does this mean? That means even though we computed this complex model on a huge data set of a UK cohort with predominantly women, the core metabolomic wiring it identified in it contains so much information that it predicts disease, or in this case, classifies disease in metabolomic cohorts that are measured on a completely different topic, completely different site. So again, this shows that similar to what I showed with the network, there's something in the blood metabolome that is so deeply hardwired that it would translate cohorts, ethnicities, diseases, and even various different blood collection processes. Some of these were not exactly best standard, and it still works. So there's something intrinsically hardwired in the metabolome of the blood that we can recover. So now we're going a little bit into the diseases. These are now less technical, more application. So we do a lot of work with big consortia here in the US on Alzheimer's disease. We have a bunch of blood data as well, but today I'm going to mostly focus on brain, similar to what Stu showed earlier. So I'm glad he introduced all of that, and then show one glimpse of blood data that we have. So one study that we're very excited about was, and if you work in the field of Alzheimer's research, especially here in the US, you might have heard about the ROS-MAP cohort. It's this cohort based in Chicago, or at the Rush, coordinated by Rush University. And the special thing about it is that, first of all, for some of these patients, they have 20-year follow-up trajectories, so very precious data. And they have postmortem brain samples, which is a precious and difficult specimen to work with. And so we had access to a new set of metabolomics data sets on about 500 of these people and their postmortem brain metabolomics, and basically did a huge correlation analysis of all these metabolites that we measure in the brain with various clinical or either post-mortem outcomes or lifetime trajectories. So basically what you see here is all the different Alzheimer's parameters we worked on. So we have pathological assessments of amyloid and tau, more combined pathological assessments, diagnosis, and then actually also cognition at the last time point before death, and cognitive decline on the way to death. So what the delta was, they're going up and down. Usually, of course, they progress down. And then did a huge correlation analysis, and I know there's a lot of data on this slide, all those metabolites, how they correlate with all of these clinical outcomes. So this is treasure trove of interpretation. So even what I'm going to show you today is just a few pieces of story that we picked. There's way more in this data than what I can show you today. It's all online. You can download it and play around with it. But sort of it paints this picture of the metabolic dysregulation in the diseased brain. And we found that over half of all metabolites that we can see are changed with Alzheimer's parameters, even though if we try to correct for medication, sex differences, and all these confounders that could lead to false associations. Just real quick, we also did some replication analysis. Very difficult because there's not a lot of studies that are here. I should say, I forgot to say this. So this is in the dorsolateral prefrontal cortex, and some of the replication was in other parts of the brain, such as the temporal cortex. It's a bit questionable. So is this really replication, or am I just checking it in a different brain region? Again, since these brain samples are so rare, it's not very easy to find in independent validation. But now I'm gonna, and again, this slide is already condensing a lot of the information into something that looks simple, that we devised just from our data. And again, this is biased towards the pathways we picked. But so what do we see in a brain, in an Alzheimer's disease brain? First of all, what's interesting is we do see a change in cholesterol metabolism and sterols, which fits with the fact that, as we know, ApoE, which is a cholesterol-based gene, is one of the, so the genetic variation is one of the main predictors of Alzheimer's disease lifetime risk. We see, and this is something that emerges everywhere, and we've even seen it in some of the talks today, we see strong bioenergetic changes. So their mitochondrial metabolism, as we can see it in these tissues, is completely changed, related to glycolysis, branched amino acids. Also, then acylcarnitines would be mitochondrial metabolism. We see, again, potentially expected, and we've heard it too, but we can actually see it directly in the metabolomic data, that you see increased inflammations directly in there. For example, downregulation of the omega-3 fatty acids and upregulation of pro-inflammatory lipids. And then there was this one group that basically we kind of picked ourselves. It's not really a pathway, but we found a lot of changes in metabolites that are known to be involved in osmoregulation, which can actually have a variety of impacts in a cell. It has to do with pressure, of course, right? Because of osmosis. So a change in osmoregulation can have effects from protein folding molecularly all the way to actually sort of spatial constraints in the skull, which is a restricted volume, right? And there's been studies there as well, and we can see all of this sort of in these brains. I want to briefly talk about amyloid versus tau, I guess, although I'm not going to take a strong stance here. What we see, and this plot is a bit complicated, but we see it both in metabolomics and also in a related proteomic data in these brains, is statistically we can try to disentangle tau and amyloid effects. And don't look at the plots too much, listen to what I'm saying, is what we see is that tau effects are not driven by amyloid as a confounder, but amyloid effects are driven by tau as a confounder. So what that means is that, at least in our data, tau itself directly correlates with the metabolic changes, whereas amyloid does not. It's actually because of tau. Now, does that mean I'm saying I'm a supporter of the tau hypothesis against it? I don't know. It's just, I can tell you that in our data, with the statistical approach, it looks like the metabolic dysregulation we observe is more because of tau than of amyloid. Also, these are post-mortem brain samples, right? So this is as late as it gets in the disease stage. But that's just my take on this topic. And now we're currently expanding this brain views across different regions. We also look at another tauopathy, PSP, which is interesting because it doesn't have amyloid pathology. So we're expanding these ideas to more data sets, more brains. Just one other last aspect of Alzheimer's, and this is still pretty preliminary, so I can't show all of it yet because it's not ready yet. So now if we go to the blood samples, biocrates P180 samples here, and we go into the ATNI study, what we see is if we cluster patients based on the entire metabolomic profile, you actually don't see much. In fact, if you see what explains why people are clustering, it's mostly cholesterol and lipid parameters, well, because that's the majority of what's in the panel. However, if you go specifically into the acylcarnitines, suddenly you get this very interesting subgrouping where you see subgroups of patients with different pathologies emerging. So why I'm saying this is also as a general lesson is if you cluster people, it kind of matters on what you cluster them on. If you put the entire metabolome in it, then it's going to be clustered by something else. And if you cluster them by a certain pathway. And in this story, we actually jumped on the train of biological age predictors, which are famous in epigenetics, but also we ran this on the acylcarnitine, so sort of a mitochondrial health biological age model. And it turns out that we can identify subgroups of patients that are surprisingly healthy for their age. So they are healthier than they should be compared to their peers. And according to our model, they're actually bioenergetically younger. And we're still working out what that exactly means, but that's sort of the latest on that front. I'm going to skip this. So now COVID. Similar to our lab was also in this schizophrenic state that we had to jump on this and work on it. I will say though, there was a lot of interesting insights in critical care, also long COVID. I'm going to show a little bit of that. So just a few slides. We had a bunch of studies. If you're really interested in this, check out the webpage, but I'm going to go through a few pieces from some of those studies. So in March and April 2020, the New York Presbyterian Hospital in New York City, which has sites in Brooklyn, Queens, and Manhattan, was what we now think was one of the hardest hit places on earth. It was a crazy time in there. If you talk to sort of all these co-authors and now friends are the critical care doctors, the pulmonologists that were treating those patients. The stories they tell are crazy. But some of them were cerebral enough in the moment to actually collect samples from this first big wave of COVID that crashed over New York City. And also a bunch of control samples that had COVID scares, but ended up being something else. But in this cohort, we had 330 patients with actually multiple time points. Unfortunately, the time points are a little too, multiple time points sounds awesome, but they were on day 1, 2, and 3 of the hospital stay. So looking back, we wish they had done it more like 1, 2, 3 weeks. So the multiple time points don't give that much information. But sort of one model we came up with that was very interesting is, so when my student Mustafa, who's now graduated and gone, was talking to the clinicians on this study, he realized that the clinicians have a very good understanding of a severity scale of the patients. They can tell you exactly on a scale from 0 to 100 who was the sickest patient they saw and who was the healthiest patient they saw. So he was thinking, how can I formalize this? Is there a way that I can actually come up with a number from the person who was the most sick to the person who was the most healthy? And together with our intensive care physicians, we came up with a scheme where basically we're saying, okay, if a person died, first of all, they are always more severe than a person who survived, obviously. And then if they died really quickly, they were more severe than if they took longer to pass away. Then on the healthy side, there are, again, categories to strategize. You can say that they need mechanical ventilation, kidney replacement therapy, and so on. So we came up with this hierarchical scheme that in the end, we have every patient from, again, the healthiest to the one who was the most severe. And then instead of sort of running models and prediction models on everything, death, replacement therapy, severity, we have just one model to fit. And with the metabolomics, we get a decent prediction power into the future. It's like 1 or 2 weeks into the future, which also replicates. Now, will these scores actually make it into clinical practice is unclear. So this is still a research result that we can predict severity and outcome. And not a translational result yet, but it would be interesting to think about if in the end we can do it with only 6 metabolites. You can get a decent enrichment of prediction of severity over your normal parameters like overweight and sex and so on. So, and then we had the study, I'm going to just stay on this slide for a little bit, where we were comparing now we're going to the very sick patients, those with ARDS, is acute respiratory distress syndrome. They are in the ICU. Death rates are between 25 and 50%, depending on which subgroups you look at. And we compared those that are suffering of COVID ARDS with those that are suffering from bacterial sepsis ARDS. So the same overall syndrome, but caused by a completely different disease. And then we did metabolomics, proteomics, lipidomics in blood. And first of all, again, I can't go through all of this. They are vastly different, even though the presentation and often the treatment is very, very similar. They're completely different in their profiles. And then even if you go into the ARDS patients of COVID, so this is the 2 groups versus each other, the 2 etiologies, 2 diseases. And now inside of the COVID ARDS patients, if you look again at certain types of clinical parameters, for example, acute kidney injury, which is a common symptom or effect in these ARDS diseases, or thrombocytosis, you will see all kinds of changes, for example, related to, again, energy metabolism, or down here we have cell adhesion and inflammation pathways. And actually some of these led to hypotheses for potential combination treatments of drugs that are already on the market for COVID. And now I want to show one case, which is now proteomics. Sorry for that, not metabolomics, but it's a really cool case of how you can do these longitudinal profiling. So, and it kind of puts it all together. It turns out So this at-risk cohort is the first one. It's the one where they came into the emergency room. If you cluster them by proteomics, there's this one subgroup that has a specific phenotype of low protein abundances, high amount of thrombocytes, also of platelet counts, and just general severe disease and a high rate of death. And they're related to these proteins that we know have to do with vascular injury. So, and you can see that in blood. And now the interesting part is if you now go to the ICU cohort, you see the same group. So again, the signature of vascular injury in blood by proteomics reappears in the ICU cohort, again, related to significantly worse outcome. Again, now we have death rates over 50% if you're in the poor group with high vascular injury. Now, one step further in the clinical trajectory, if you go into pathology, so this is now from postmortem autopsy samples, again, you see that there's this subgroup that has high platelets and strong markers for these vascular injuries. So it goes all the way to death. Or maybe on the brighter side, but still significant, if you now go to those patients, we had data for some that were discharged from the ICU, went home. So if we now take those that when we took the sample had this bad signature, the vascular injury signature, and you follow them up 12 months later, by this recovery scores, is basically asking if they can do everything on their own, if they have pain, if they're okay sort of in life. Then you can see that those that originally had this vascular injury phenotype when they were in the ICU have significantly worse also recovery later on. So it's this sort of one aspect. It's probably not everything vascular injury, not by any means, but it's this phenotype, this molecular phenotype that goes all the way from when they come into the hospital through severe disease, home, and then still they have to deal with this later on, and it's all related to this vascular injury. Okay, so now I'm going to briefly talk about cancer, so kind of clean slate, new topic, and then the dog one is actually also about cancer. So, and here again, I'm going to talk about blood. We also have some tissue stuff. I'm going to show it one slide in a minute. So one interesting story here was, and this really was for me still one of the most fascinating findings of all that I have had in the metabolomics case. So if you take follicular lymphoma patients, it's one of the non-Hodgkin lymphomas. The other famous one is diffuse large B-cell lymphoma. And we predict event-free survival. So until the disease progresses or the cancer gets worse, then there's a single metabolite. So it's not a signature, a score, no machine learning. It's just one metabolite, N-formylmethionine, that whether you're high or you're low, predicts actually pretty well whether you're going to progress or not. So why this is interesting is, first, if you look at the x-axis here, this is years, right? So 3,000 days, we're somewhere here at the 10-year mark. And follicular lymphoma is considered to be an indolent disease. This is a really slow-growing lymphoma. These patients are not under chemotherapy initially. So they're just waiting and watching. And nevertheless, they have something in them that shows up in blood that already very early on tells us they have a different type of tumor, and this might actually be at a higher risk of progressing later. It also replicates pretty well. So what we then did is we actually went to an immuno-based assay because you only measure one metabolite cheaply. You can do it in many samples. No mass spec needed anymore, and it validates in a replication cohort. And sort of why that's interesting is what I want to sort of talk about the slide for a second is whenever we talk about blood metabolomics, I think we heard earlier, this might be hosts, this might be, where does the metabolite actually come from, right? Why is it in blood? Did it come out of the cancer? Is it a reaction of the body to the cancer? Is it a lifestyle change of the patient? Maybe they started eating something else and that's why it's in there. So that's actually a very difficult question. And in every single case, you have to do your own research, additional research to figure it out. However, this particular one that I'm showing you here, we actually, with a lot of additional research where I don't have slides yet because they're still in the middle of being generated, we know it's coming out of the cancer cells. We actually know it's coming out of the mitochondria of the cancer cells. And this is a very specific, very well-regulated mechanism of mitochondrial stress. In fact, it's not even N-formylmethionine, the amino acids, it's N-formylated peptides. It's just, it broke off in the mass spec and we measure the N-formylmethionine. And it's a very well-regulated complex mechanism that the cancer cells have early on with mitochondrial stress that later on leads to a high chance of it progressing into an aggressive disease. So it's a super cool story, but the point I want to make is that it's, now that I put it on the slide, it's maybe easy to understand, but among hundreds and hundreds of metabolites that might be changed in blood, figuring out which ones are actually coming out of the cancer is often very difficult, and this needed cell lines, mice, additional data. So there was a lot of work going on until we were confident to say this comes out of the tumor cell. So much for this just also what blood metabolomics does. And then just one more thing I wanted to show. This is very recent. So now we're going into the tumor, not single cell, because single cell metabolomics is maybe up and coming, but it's going to be definitely difficult if you think about mass spectrometry. But tissue-based samples are still interesting. You know, you have a mix of all the bulk cells together, but it's decent enough to analyze. And what we realized a couple of years ago is that the TCGA, the famous public cancer atlas, has no metabolomics data in it, right? So what at Resnick, he actually started this, is he basically picked up the phone, or more like emailed, but also called some, and tried to get every cancer metabolomics tissue data set out there that he could get his hands on, including also always with matching transcriptomics and sometimes with this tumor normal thing. And we put it all together into what we call the CAMP. This is a pun, of course, for cyclic AMP, or the Cancer Atlas of Metabolic Profiles. So we now have this resource of about 1,000 samples of various different cancers, metabolomics, transcriptomics, that you can do a lot of analysis on. One big disadvantage, and this is similar to TCGA, is of course the clinical data for all of these different cancers are not very homogeneous. It's very difficult. So what we did in this paper was we tried to find some big pan-cancer signals across these different data sets. And I'm just going to show you one of the analysis that we thought was fascinating. So if you think about transcriptomics and metabolomics now in the multi-omics context, and you're already in a tissue, so if you look at transcripts and metabolites that correlate with each other and ask yourself, what are those? Who correlates with each other in metabolomics and transcriptomics? Your first thought might be, well, it's probably something like enzymes and their products, right? So you have a metabolite, you have the enzymes transcript. Those should somehow correlate. Or maybe the metabolite in a transporter. That seems logical. Turns out that's not at all the case, even though we correct for confounding factors. So in fact, out of those that significantly correlate across cancers, metabolites and transcripts, only a fraction, 2.5%, are explainable by these this is the direct cascade of how the genes work with metabolites. And the vast majority is not. And that was a truly surprising finding. Maybe it was too simplified what we assumed, that it would be these individual cascades, but it's really not. So the question is, if 97.5% of correlating transcripts and metabolites in tumors are not individual pathways, what is it? And then what we found in this study is it's probably predominantly driven by immune mechanisms and also correlated to immune cell infiltration. So in fact, a lot of the metabolites that correlate with a lot of transcripts are related to immune processes in these cancers. And that seems to be the main driver of the connection between gene regulation and metabolism in these cancers, not the tuning of a single pathway step in glycolysis or so. That's not the main driver of these gene metabolism connections. All right, now the last one, again, the pet project. These are actually pets. So the Cornell has a very active veterinary clinic. They're specialized on all kinds of animals, and they have a good biobank, and it turns out they have lymphoma patients. Also, the original discussion was if we can collaborate on Alzheimer's disease. Now, cognitive assessment in dogs is actually possible. The tests are kind of interesting. It's, of course, you can't do the same thing as in a human, right? You can't do regular cognitive assessment like the tests, but you can do it if you have an old dog. It's actually interesting to try it out. For example, one of the questions is, does your dog stare against the wall a lot? And then if that's true, then that's actually a sign for cognitive decline in the canine patients. Now, cancer is a little easier because you can take a tissue and analyze it, right? So we don't have to think so much about behavior. So what we did is, and this is actually funded by some philanthropists, we already had, similar to the follicular lymphoma that I showed earlier, we had a matching cohort with diffuse large B-cell lymphoma, human blood samples, control versus cancer. And now we did the same thing in dogs. So we were working with the nurses there. They walk into the operating theater, they get the blood samples and the tissue samples. For now, we only focus on blood, do metabolomics. And now we start, and this is very early data. This I copy pasted from a slide also a couple of days ago. So we're trying to compare what is the blood signature of the disease in the human versus the canine. And the answer is it's very heterogeneous. So there are some pathways where the human and the dog system agree completely. So they're up and down together. And then some pathways where they completely disagree. So they go up in humans and down in dogs. And we're currently trying to make our way through this data. So the long-term promise of these types of projects is that the humans might help the dogs. Because again, these are not lab animals, these are pets, right? So the humans might help the dogs because, of course, lymphoma research in the human system has a billion-dollar industry behind it. So if any of these drugs might work for the dog, you might see it in such a data set here, or at least get an indication. And actually the dog might help the human because these kinds of diseases happen way more quickly in the dogs. So a dog trajectory of cancer is probably a year or less, and you've seen everything, which is of course terrible for the dogs and their owners, but very interesting in terms of the completeness of clinical data. You get a lot of fast clinical data from these animal models. And with that, I want to thank all of these people from the different cohorts, and I hope it wasn't too much content today. I just tried to give you little pieces from all the different things we do, and thanks for listening.